Hi everyone, and thank you very much, Lorenzo, for agreeing to give a Build a Cell seminar, and you can get started.
Thank you, Kate, for the invitation. It's always great to participate in this great series. And today I'm going to give everyone a little bit of an update on some recent and also not so recent work we have been doing in the group. Not quite about building synthetic cells, but rather about building synthetic organelles that you might want to use as components for your synthetic cells. And we're going to build those organelles out of nucleic acids and in particular RNA. Right. So biomolecular condensates, I'm sure that many people in the audience will know exactly what we're talking about, but for those one or two people for whom this is perhaps new. So these are little droplets that exist in cells. They exist in all kinds of cells, from bacteria to plants to mammalian cells, and they are made typically of proteins and RNA. I say droplets in the sense that people believe that in most cases these emerge due to liquid-liquid phase separation, so like oil in water type behavior. And there are many different types of them, dozens in fact. Some of them have been known for decades, like the nucleolus. Others people keep discovering all the time. And in a eukaryotic cell, for example, they will exist all over the place. They exist in the nucleus, they exist in the cytoplasm. They have different compositions, they have different names. We're not going to enter into that. And they play different roles. And they generally regulate cellular organization and biochemistry. And they function by recruiting or excluding molecular clients, therefore changing local concentrations. And if you do that, you can then regulate enzymatic processes, metabolic processes, and biochemical processes in general. What we're trying to do here, and what a good chunk of the research going on in my group is about, is trying to build designer condensates or synthetic versions of these condensates that we can then use to engineer cells and synthetic cells. But more about this, why do we care about creating synthetic biomolecular condensates? Usual aim here is that of learning about biology. So if you can create synthetic versions of these droplets, you can learn about the biological ones, ask interesting biophysical questions. Perhaps the greatest focus that we have is about using these in the context of synthetic biology, so engineering synthetic cells that I don't need to expand on for this audience, but also create synthetic organelles within a living cell. So say a hybrid top-down bottom-up approach to synthetic biology. And there could be, and there are, applications of biomolecular condensates as materials in their own right. So you can use them, for example, to control industrial biocatalysis, colocalizing enzymes to make them more efficient. There are people who are interested, for example, in using them as vectors for drug delivery, among other applications. So there's many good reasons why you might want to engineer synthetic condensates. How do you go about doing this? Now, we're not the first to try and do this. There's a vast literature. People have been trying and managing successfully to engineer synthetic condensates for a long time. And most of the implementations that you will find in literature rely on synthetic proteins and peptides. So the key ingredient that you need to create these liquid droplets is a multivalent molecular building block. And with this, I mean a molecule that can bind to a few other molecules relatively weakly. So these are weak multivalent interactions. The way in which you can achieve this, let me get a laser pointer here. In protein building blocks is generally one of two ways. So one of them, which is the most common one, is to use intrinsically disordered domains or intrinsically disordered regions. So these are disordered beds of the protein that interact in many ways, for example, through pi stacking, electrostatic interactions, hydrogen bonding, and these many weak interactions promote phase separation. Or you can have slightly more engineered designs in which you have specific stickers spaced in some way by some flexible non-sticky regions. Either way, there's a huge literature on this. This is a very nice review, but there are also newer ones. And there are many advantages in using proteins and peptides to engineer phase separation. These are very functional, they are stable, they're responsive to stimuli. We are learning more and more using machine learning how to program and engineer proteins. And very, very importantly, these objects, they can be genetically encoded. Therefore, they can be expressed within your cell or within your synthetic cells. And that's a big advantage. But there are also disadvantages. So here, if you read the reviews, you look at literature, it sounds easy to control the properties of these condensates with proteins, but actually it's not that easy. And the reality of things is that while, for example, using machine learning, we can predict quite well the structure of proteins that are well folded and compact, it's not quite that easy to predict the behavior of these flexible and disordered proteins. And therefore, it's quite hard to say, prescribe the properties of your condensates and then design a building block that will give you exactly those properties. So there's still a significant bottleneck when it comes to programming condensates with proteins. An alternative approach, which is what I'm particularly keen about, is to use nucleic acids instead of proteins as building blocks. And you might want to use DNA, and this is one of the applications of DNA nanotechnology, which again, I'm sure many people will be aware of. So this is a concept and a field established by Ned Seaman around 40 years ago. Sadly, Ned passed away a few years back, but he had this incredible idea that it might be possible to use nucleic acids starting with DNA, not just as carriers of genetic information, but rather as building blocks to create nanostructures and devices from the molecular scale all the way to the large nanometer scale and even larger. And you can make little tiles out of DNA, little junctions. You can use DNA origami to build more and more complex structures, all the way to bunnies if you are so inclined. You can build synthetic condensates using DNA. The simplest way to do this is to create a DNA junction. As you can see here, this is the basically elementary element of all DNA building blocks. So this is kind of similar to a holiday junction for the biologists among us, but it's a fixed holiday junction, so it won't branch migrate and it will not slide. And then you can attach or add at the end of each arm of this junction some sort of sticky moiety. The simplest thing you can do is to just have a little bit of single-stranded DNA that you can call a sticky end. And if you have 4 sticky ends, you can design them such that they are self-complementary. Then you suddenly have created a tetravalent particle. So you have this multivalency, and you can make this interaction sufficiently weak such that these nice tetravalent particles undergo liquid-liquid phase separation and form these very nice droplets, as you can see here. So that's the simplest thing you can do. Here's an excellent review by the group of Omar Saleh, summarizes quite a lot of the biophysics of these systems. So we have made our little contribution to this particular strategy, introducing a variation on the theme of these DNA junctions that people call nanostars by replacing these sticky ends with hydrophobic molecules that by and large they behave in the same way. They bind to each other hydrophobically instead of having a selective base pairing. And depending on how you treat your system, how you design your nanostructures, how you anneal it, this actually might form solid condensates in a way, solid crystals, as you can see here, or they might form amorphous or liquid droplets, as you can see here, in this case with a faster anneal. So that's not something I want to go much deeper in, but I just want to give you an overview of quite a lot of the functionalities that it's possible to create with this synthetic DNA condensate. So now these results are just from a few years or several years of work in my group, but there's out there a lot more from others who contributed significantly to this research. So for example, you can control the molecular level structure of these synthetic DNA condensates. This is actually for the case of the crystals. These are X-ray diffraction patterns. You can see that these peaks shift, indicating that you are changing the periodicity of the lattice. Therefore, you're changing the porosity of these synthetic DNA crystals in this case, but it applies the same when it comes to the amorphous or liquid-like condensates. You can control the size of the condensates, going all the way from micron or cell size down to the nanometer scale. So you can make nanoparticles out of these condensates that could be useful for drug delivery. You can engineer assembly and disassembly subject to many different stimuli. For example, exposure to certain molecular triggers like nucleic acids, changes in ionic conditions, changes in pH, temperature, and so on. You can create interesting patterns in these condensates, so you can basically create phase-separated domains within the phase-separated droplets, and we're going to expand on this a bit more, and this imitates characteristics of the natural biomolecular condensates. You can establish dynamical phenomena within the condensates, so say molecular reaction networks that take place within them, as well as localize enzymes and biocatalysis and use the condensates to spatially organize biocatalysis a little bit as it happens in natural condensates. So this was, again, an overview of many years of research in just one slide, but the point I wanted to make is that these synthetic DNA condensates are actually quite cool. So I don't think they are any less good compared to proteins when it comes to engineering functionality. They're quite stable, they're responsive. Importantly, they are way less complicated than proteins to engineer, so this is really an exquisite example of rational design, I believe. You can go from the nanostructure that you want, to the function that you want, at the condensate level, to the sequences that you need to create those nanostructures and those functions in a quite robust way, aided by some computational tools. So these are very cool, and I think they tick a lot of the boxes of what we might want to do with the synthetic condensates, but they have one critical drawback, which is that they cannot be expressed. So maybe some of you will tell me otherwise, but I don't know how to teach a cell to build a DNA nanostar with cholesterol molecules attached at the end of each arm. So that's tricky. As far as I know, it can be done. And that's a huge drawback when you compare the protein implementations with the DNA ones. So how do we address this drawback? Perhaps we can move one step up our central dogma ladder and go from protein, from DNA implementations, not quite all the way to protein implementations, but rather stop in between and try with RNA nanotechnology and RNA-based condensates. So now, RNA nanotechnology is by and large similar to DNA nanotechnology. You use now synthetic RNA molecules, make them fold into nanostructures with a prescribed shape, size, and interactions, and it works not quite as robustly as the DNA approach, but let's say almost. There is one key difference that we're going to expand later on, which is basically the point of RNA nanotechnology, is that you want these nanostructures to actually fold co-transcriptionally. So you want to encode this RNA into a gene, then transcribe this gene, and you want your RNA molecule to fold while it's being transcribed, therefore isothermally. Because of this, the way in which you design these nanostructures is a bit different, and most often you will form nanostructures that are actually folded from a single molecule rather than being composed of many different molecules that will anneal together as you do with DNA. That's because with DNA you have the luxury of being able to heat up and slowly cool down your system in order for these molecules to assemble together in the right way. With RNA, again, you want to do this isothermally and co-transcriptionally, so you don't have this kind of luxury. But the hope is that with RNA nanotechnology, we will have the advantages in terms of programmability, functionality, responsiveness that we have with DNA. They remain less complicated and more easy to engineer and to have our rational design angle compared to proteins. And the key advantage is that they can be genetically encoded and expressed. So we can, again, put our building block into a gene, put it into our synthetic cell or into our biological cell, and then we're going to make loads of these RNA and hopefully create our condensate. So hopefully with this approach, we can retain all of the advantages of DNA condensates and reject the disadvantages. So this was a lengthy introduction, taking us to the goals that I would like to try and achieve in this talk, or at least tell you about how we managed to achieve these goals. And I would like to address three questions. So first, can we actually build our designer RNA condensates? Second question, can we use them to imitate interesting properties and functions of natural condensates. And finally, the most important question is, can we actually express them into living cells? So let's start with the first. And the answer to all three is, of course, yes, but we need to see how this is done. So let's start with the first question. Can we build design RNA condensates? The way in which you do this is by creating RNA versions of our DNA nanostars. So here is one such designs. The key difference, really, if you compare with the DNA-based implementation, is that now this is folded from a single strand. It's not assembled out of multiple strands. You have one strand going around, forming this junction. Now, because this is a single strand, you can't have little DNA overhang or sticky ends that requires multiple strands. Therefore, the way in which you induce affinity between these building blocks is by having these domains that we call kissing loops. So these exist in biology. For example, the HIV RNA has them, and they are shown here. So they're palindromic. So each of these will bind to identical loops. Therefore, once again, you have a pretty ideal tetravalent particle made out of RNA. The other important feature here is this MGA, which stands for malachite green aptamer. So this is a fluorescent light up aptamer. So it's a little bit of DNA that has been selected such that it binds to a bad fluorophore. And when the fluorophore binds to it, this becomes a good fluorophore. And therefore, you can see your RNA being made. Then all that you need to do is to encode this in a certain bit of DNA, which can be linear or circular under T7 RNA polymerase promoter. In this case, we're going to do an in vitro transcription reaction. You form loads of this RNA, and you see that eventually you get nucleation of big droplets, coalescence, they grow, they are quite large, larger than a cell. Eventually, they will start degrading, so they form condensates. So this we call the A design. We can have different designs. This is the B design, which is the same as A, but with different kissing loops, which are now orthogonal to A. So as we're going to see, B binds to B, A binds to A, but B doesn't bind to A. And also the aptamer is different. This is called the broccoli aptamer. So these are often called like vegetables. And it's just a different color such that we can distinguish them. But otherwise, they behave very similarly. We have a third design that we call C, and this is just to show you that actually you can have different physical properties of the condensates. So C has a different kissing loop compared to A and B. Again, it's orthogonal to A and B, but it forms condensates that are actually more viscous, and they fail to relax into these discrete droplets, rather forming this gel-like network that takes a long time to coarsen and relax. And this one has both aptamers, the broccoli and the malachite green, such that we can distinguish these constructs again from A and B. I won't bother you with much of this, but you can do all of your characterization, make sure that your RNA is transcribed properly with some electrophoresis. You can look at the physical and rheological properties of these condensates. One important parameter that people often measure for these materials is something called inverse capillary velocity that you can deduce from the rate at which two of these droplets coalesce with each other, and you can get some numbers that are sensible when you compare them with the huge range of rheological properties that you have in the biological condensates. I mentioned earlier that A and B are orthogonal to each other. You can demonstrate this by co-expressing again in vitro A and B. And you can see that you get the red A condensates that coalesce with red, the cyan B condensates that coalesce with cyan, but they don't coalesce with each other. They remain completely demixed. And here is an array of samples in which we change the relative abundance of the templates that code for A and B, therefore the amount of the nanostars of each kind. And if you do this, you can change the relative size of the two types of condensates. You can also express them in synthetic cells, the very simplest version of which are water-enol droplets that contain your in vitro transcription machinery, your genes. And if you let this reaction run, you see that soon each of these water-enol droplets will form exactly two condensates, one cyan and one red. You can zoom in and see that also in this case, you can change the relative size of the cyan and the red condensates by changing the abundance of the gene that codes for the A and B nanostar. If you want, you can have three such synthetic organelles in your synthetic cells, A, B, and C. You can also make them into genitourinary vesicles, more cell-like types of synthetic cells. It's still unpublished, but hopefully will be published soon, and it works pretty much in the same way. So we can engineer synthetic cells to express their own RNA-based synthetic organelles. We can have different numbers of them and control the relative size too. So we can tick the first question off. So we can design and build synthetic RNA condensates. Can we now use them to imitate some slightly more interesting characteristics of the natural condensates? And natural condensates have a lot of very interesting and complicated characteristics that we need to build towards with our synthetic replicas. So one of these characteristics is the presence of internal phase-separated domains. So many condensates, and the typical example is the nucleolus, are not uniform. They have internal pockets that have different composition from the surrounding phase or from other subdomains in the condensate. And again, the nucleolus is the typical example because it has up to 3 nested compartments that sequentially participate into the synthesis and processing of ribosomal RNA. So the nucleolus basically makes ribosomes, and you can see this, and then this, and then this. And this kind of coexistence is really, really important for making this process of generating and modifying the ribosomal RNA efficient. So the question is, can we create or can we engineer internal phase separation in our synthetic RNA condensates? A good place to start is to start from our non-miscible condensates A and B that you can see here, and then introduce a third component that we call a chimera or linker nanostar that now has 2 kissing loops that are red, so they bind to A, and 2 kissing loops that are cyan, therefore they bind to B. And the idea is that the more of this you express, the more you go from a situation in which A and B are demixed towards a situation in which A and B are fully mixed, because these linkers basically help the A and B phases mix together. And in between, hopefully we'll get situations in which you have these partially mixed condensates that have domains that are rich in A as well as domains that are rich in B. And if you do this, here from left to right, again, you are increasing the amount of the gene that codes for the linker, you get exactly this. So here on the left-hand side, you have a collection of droplets that are basically pure A and pure B. However, these are glued together by a little bit of linker that you are expressing. On the opposite side, you have situations in which the linker is so abundant that basically it induces full mixing between the A and B phase, therefore you no longer have separation and you have a single phase. And in between, you get this quite rich array of different shapes and structures in which you have partially mixed A and B domains. So effectively, we have created these multi-compartment condensates. So now there is quite a lot to say about this, which I'm going to only very, very briefly touch on. And the important point is that the shapes of these objects are really weird. So this one is kind of hollow things. This one has this central blue domain with then some red and then some other blue outside. This one looks like the Pokemon Squirtle, so we call it the Squirtle face. But the point about this is that these are strange. So if you consider a very simple system in which you have just 2 semi-miscible liquids existing in a background of a third liquid, depending on what the interface tensions between the different components are, you expect to get some interesting morphologies. You can get Janus droplets such as this, partially engulfed states such as this, or fully engulfed states such as this. But you don't expect to have the Pokemon phase or hollow capsules or this slightly strange, we call them sunflower type condensates. So the hypothesis here is that these interesting morphologies actually emerge because the condensation process here occurs very much out of equilibrium. So once again, here we are expressing our RNA isothermally. While the RNA is being made, eventually these condensates start to nucleate, grow, coalesce, merge, and rearrange in some way. And this leads to these very complicated morphologies. And indeed, if you take, now this is a much bigger phase diagram in which we change the relative amount of A, B, and the linker 10 testing different compositions compared to the simplest picture I showed earlier. But what we have here on the left-hand side is the result of, say, co-transcriptional assembly of the condensates, and then you take all of these samples and you heat them up and cool them down, which should help them get closer to thermodynamic equilibrium, and you see immediately at a glance that the structures look different. So this is to indicate that probably it is the case that these quite complicated condensate morphologies are the result of non-equilibrium assembling. We are looking into this in more detail, and hopefully we can report on it sometime soon. The second interesting property that we wanted to demonstrate with these synthetic condensates is the ability to capture proteins. Now, it's all well and good being able to control the structure and rheological properties of these condensates with nucleic acids, but ultimately, if you want them to have some interesting functions, you need to be able to recruit proteins within them, because in cells and in synthetic cells, proteins are the carriers of interesting functionalities. It's very easy to capture proteins within these RNA condensates. All that you need to do is to consider a protein that can be targeted by an aptamer. In this proof of concept example, you have YFP and you have an aptamer that can bind the YFP, so that's an RNA aptamer. Then you can engineer your building block with an overhang that is complementary to another overhang on your aptamer. Then you co-express your condensate building block, the aptamer that binds to it, and in the presence of YFP, you can see that the YFP gets captured within the RNA condensate. If you omit the aptamer, the YFP will remain dispersed. You can try this with different proteins as well. In this case, it's with streptavidin, and it works in the same way. These are the ones that are published, but now we have a few more examples of this working nicely. So we can take off the second question as well. We can use these RNA condensates to imitate some interesting behaviors and structures of natural condensates. And finally, the most important and perhaps interesting question of them all, which is, can we now leverage the fact that these building blocks can be genetically encoded and express them actually in living cells to create synthetic organelles within our living cells? And this works quite nicely. So because we like simple things, we start from the simplest cell to work with, which is E. coli. You clone your gene, say for the A nanostar, into a plasmid. You need to introduce a terminator domain. We change a little bit the design of the A nanostar. We switched the malachite green aptamer because the dye of it is toxic for the cell, so they don't like it, with another aptamer called pepper, which is well tolerated. And then you put this into the cells. And we have two different designs. So A, which again is very similar to the one we have studied in vitro, and this A bar, let's say, which is the same as A, but we changed the sequence of the kissing loop such that they no longer bind. This is very useful as a control because you want to demonstrate that the condensation of these building blocks is not just due to overexpression of RNA and RNA crashing out, but it's specifically due to this base pairing at the kissing loops. You can transform the cells, you can induce them, and you will see that when you express A, you have the formation of beautiful condensates. Pretty much each cell has them, and they tend to sit at the poles of the cells. And you can see this by eye, but you can do some image analysis, taking images of all the cells, registering them and averaging them, and then you get these fluorescent maps where these bright spots indicate where the condensates tend to be. This is just a statistics of how many condensates there are per cell. You can see the legend down here. Most cells will have exactly 2 condensates at the 2 poles. Some cells will have 3, and we'll come back to that. But the important point is that if you do the control with the scrambled kissing loops, you get uniform fluorescence, indicating that this is not just random RNA crashing out and being pushed at the poles, but it's actually condensation specifically induced by the base pairing. You can do the same with nanostar B, and pretty much it works in the same way. There's some interesting things to notice. The condensates might be localized at the poles, but it doesn't mean that they form at the poles. In fact, if you look at videos of these cells expressing the condensates, you can see that quite often they form little droplets in the middle. You can see next to my laser pointer, and then these droplets get pushed towards the poles. So you might be wondering why. these condensates are pushed towards the poles of the cell. We think that the main reason is that the center of the cell is quite crowded because it contains the chromosome of the cell, and you can do a very simple back of the envelope calculation. People have measured actually the pressure exerted by the presence of the chromosome in the center of the cell. That's this value here from this very nice reference, and you can work out, given say a condensate of 100 nanometers in radius, which is probably something like this thing here because you can see it with the microscope, what kind of energy cost it would imply forming such cavity in the middle of the chromosome of the cell. And that's quite a large energy cost. And we think that this is therefore sufficient for the condensates as they're formed to perhaps be pushed out towards the poles. There could be other reasons why the condensates accumulated the poles that people have pointed out, and I'm happy to discuss them if you are interested. Another interesting thing to notice is what happens when cells divide. So you might remember from a couple of slides back that you have that some cells have a third condensate, and not just two at the poles, but a third one as well. And we think that those are cells that are approaching division. As the cells approach division, you tend to see a third fluorescent spot forming in the middle. We believe that's because where the space has emerged due to the fact that the cell is dividing and there's no longer the chromosome in the middle. And then the cell will divide, and quite often this center condensate will be split between the two daughter cells, but in other situations it will end up all in one and none of it in the other daughter cell, but we haven't characterized that in great detail. Another important point to discuss is actually what happens in terms of degradation. So people have pointed out that RNA is degraded quite quickly in E. coli. In fact, the half-life of mRNA in E. coli is roughly 10 minutes. So the question is why these condensates exist and are stable at all, where they're not simply, the RNAs are not simply degrading as they are formed. And we have done characterization on whether the RNA is enzymatically degraded and processed. I'm not going to enter into the details. You can have a look at the paper, but it turns out that the RNA is being degraded, is being cleaved at specific locations, predominantly by cellular nucleases. But the important point is that this is not necessarily say, killer for your condensates. What we believe that is happening here is that we have a steady state in which RNA is produced at a certain rate, RNA is degraded at a certain rate, and at steady state you get a sufficient concentration of your condensate-forming RNAs to sustain the phase separation and the formation of the condensates. And you can verify this with a very simple back-of-the-envelope calculation. Once again, you can link the steady state concentration of your nanostars, which is this, to the tau, which is the lifetime of the nanostars, how long it takes for them to be degraded, and then the copy number of the plasmids, so the number of genes that you have, the transcription rate of the T7 polymerase, and the length of the transcript. These are all known parameters. The number of nanostars at steady state, you can either measure it with qPCR, as we have done, but you can also guesstimate it given the size of the condensates. You get very similar numbers. And this allows you to calculate the lifetime of your nanostars. And you get something like 20 minutes, which is very similar to what you get for messenger RNA in E. coli. A bit longer, one might argue, because these are tightly folded structures, so perhaps a bit harder for the nucleases to digest them, but it might just be a similar number. And the difference between this number and what you expect for mRNA might just be within the error. But the point here is that this makes sense. So given a sensible degradation rate for RNA and a known transcription rate and formation rate of the nanostars, you get a picture in which you can make enough nanostars for the condensates to be indefinitely stable. You can express two condensates within the cells, A and B, at the same time, as you might expect, and as we have verified in vitro. A and B still do not mix within the cells, and they form coexisting condensates. You have a red one and a cyan one here. Again, there are some interesting considerations to make when it comes to the location of the condensates and the relative size of them but this, I'm afraid, would be a bit too much detail for this talk. We can capture proteins within the synthetic condensates in the cells, pretty much similar to what we have done in vitro. The only difference is that here our aptamer, that in this case captures GFP rather than YFP, but it's a very similar aptamer, is not attached as an external appendage to your DNA building block, but rather is embedded within the arm of the nanostar. We believe that this makes it removes the extra step that you need to work of the aptamer and the nanostar binding. So when you express this aptamer containing nanostar in presence of GFP, you can see that the GFP gets captured within the condensates. It localizes very well with the condensates, and pretty much all of the condensates have GFP, and all of the GFP aggregates overlap with the condensates. And if you do a control in which you have the condensate that does not contain the aptamer that binds GFP, then the GFP remains dispersed, again indicating that this is not a case of the protein simply being overexpressed and forming inclusion bodies and crashing out, but rather it's specifically captured within the condensates. You can play some interesting games. For example, you can melt your condensates as you would in vitro. So I haven't showed this, but you can actually show that if you heat them up, as you would expect, because this is all base pairing interactions, eventually they dissolve and then they reform if you cool them down. And this works in the cells as well. You need to heat them up quite a lot, and the cells are not that happy if you heat them up to 60 degrees, but you can see in this sequence of images, as well as in this composition of all of the color maps showing the distribution of the condensates, that when you reach about 60 degrees, the fluorescence becomes diffused, indicating that you have melted the condensates. And if you cool back, they will reform. You can do the same with nanostar B. And you can show that when you do this and you have GFP captured within the condensates, the GFP will be released. And if you cool again, the GFP will be captured again. So this takes us to the end. We haven't yet gotten to the point of using these condensates for some cool applications. We're working on that, but we can definitely express them within living cells and show control over the distribution, the existence of two different condensates, and the ability to capture proteins. So in summary, we have introduced this new platform for engineering synthetic RNA condensates that we think leverages the advantages of nucleic acid nanotechnology while having this critical advantage of being genetically encoded, and therefore you can express it in your living and synthetic cells. We can program the size, number, morphology, and mixing state of the condensates. We can have multi-phase condensates. We can have protein capture. We can make them into cells. These are the people who did the work. Giacomo is the person who introduced the system in vitro, did most of the work I showed in the first part of the talk, and then Akhtar and Andre have continued some of this work, and Brian, Catherine, Milan, and Adam have done the work in the cells. So thanks to everyone in the group, all of the collaborators, funding, and you for your attention.
Thank you. What's the story with the crowns on your picture?
That's a very British thing. That's when you do Christmas dinner, you get these things that you pull called crackers, and they go bang, and inside they have little crowns, and then the British people put them on their heads. I don't know if there's any other British people in the call, but this is fairly standard.
Congrats. Yeah, this is amazing amount of work. There are some questions in the chat. Can you see chat?
I'm sure I can. Yeah.
If you can read the question before answering, because people watching the recording don't see the chat.
I will. Right. So iPhone asks, exciting method. Do the condensate sequester cellular RNAs? We don't know. What we know is that we can engineer them to sequester mRNA, and we are working on this, and we have some preliminary data that show that they can. But kind of endogenous RNAs, I would say maybe. I think it's definitely the case that they will partition RNA binding proteins that exist within the cells, and maybe that can bring in also endogenous RNAs. Lindsay asks, great talk, thank you. Can you make synthetic condensates that then make their own membrane by enzymes capturing the condensates? That would be super cool. Synthesizing a membrane. I know there are very good people who work on that. I think that would be a little bit challenging. But what we can definitely make is synthetic condensates that are externally coated with a membrane. So in fact, we have at least one paper, I think, with DNA condensates that can be covered in vesicles and forming a sort of a membrane. Expressing a membrane, it's a little bit more tricky, but it would be very cool. Another iPhone, I don't know if it's the same iPhone as before. Have you tried to form condensates in eukaryotic cells? Yes. So there is actually a paper from a collaborator, Elisa Franco, who has shown this. It's a very nice paper that came out maybe 2-3 months ago. There's another group as well who has demonstrated this, and we have some data on this as well. So it works well. You can express them in the several cell lines have been tried, X cells, HeLa cells. So yes, this can be done. It works less robustly compared to bacteria. So in bacteria, you get pretty much all of the cells that make the condensates. In eukaryotic cells, it's a bit more hit and miss, but it works. Alexander asks, can you dissolve condensates by expressing a short hairpin I guess competing with the kissing loop interactions? Potentially, yes. So there is one paper, I think by Elise as well, showing something like this in vitro. Whether you can do it into cells, I don't know, but potentially yes. If you make enough of something that competes with the kissing loops, eventually probably you will dissolve them. That's a great idea. Davis asks, thanks for the talk. Can you take the condensates? Can you make the condensates form and dissolve repeatedly in response to small molecule or light stimuli? So for the RNA condensates, we have not achieved this yet. You could potentially use light responsive peptides to mediate the affinity between between the RNA condensates and do that in cells. That would be very cool. For the DNA condensates, because you can leverage chemical modifications, then you can do all of this and more. So people have demonstrated that these DNA condensates in vitro can assemble in response to certain cations, antibodies, and you can use, for example, azobenzene modified oligos to make them light responsive. And I'm sure I'm forgetting some of these very cool results. That there's a vast literature, and this can be done definitely, and can be done repeatedly. And then Jaiki asks, can I ask what is the cation ion partition preference, for example, Mg2 plus or K plus? So I guess whether you're asking whether magnesium or potassium partition within the RNA condensates, we don't know. So in the in vitro transcription reaction, there isn't a lot of monovalent cations or at all. There is magnesium because magnesium is needed for the transcription, is a cofactor, and we know that magnesium binds to nucleic acids quite strongly, so definitely will be partitioned within the condensates. We haven't looked specifically at how they respond for the RNA ones to having more monovalent cations. Tan Mei, you mentioned about the dynamic equilibrium between RNA synthesis and degradation. I am wondering whether the synthesis timescale is enough to establish such an equilibrium. So thanks for the question. So that's the very simple calculation that I showed in my slides. I think it works out that it should be enough to establish such equilibrium. I mean, you might argue that the fact itself that the condensates exist tells you that you must be able to form them at a rate which is sufficient to maintaining a steady concentration. But again, if you consider how many condensates, how many nanostars you have in a cell, which you can measure, you can estimate, given the size of the condensates, and you know what the transcription rate of T7 RNA polymerase in E. coli is, you can find that online. You can work out what the lifetime of your condensates or of your nanostars should be, and that turns out to be in the order of 20 minutes. So that's, again, not unreasonable given the known lifetime of mRNA in E. coli. I think those are all of the questions in the chat.
I think so. Thank you. Yeah, I'm looking. Yeah, there's someone, Sam is raising his hand. Do you want to unmute
Hello.
Sam and ask?
Hi, Lorenzo. Yeah, great talk. It's very nice to see
How are
the
you?
work. And very good. How are you? I have a question also about this dynamic steady state model you showed. This, of course, assumes that the waste is not interacting with your droplets at all. Do you know that when you degrade the RNA, it can no longer bind? It can't passivate the droplets? Yeah, what do you know about the degraded RNA?
That's an excellent question. So you can see what the degradation looks like in here, or you can see in the gels, but probably you would have to stare at them for a while. But what we have done here, and thanks for the question, is nanopore sequencing of these transcripts, and it tells you exactly what the sites are where these things are truncated. And this is for nanostar A, but we have the same thing for nanostar B in the paper. You can see that they basically tend to get truncated, at least this design, at some location in the arm. Therefore, what we think is happening here is that you have actually a good fraction of these partially degraded transcripts that are not completely useless in a way, but they just have a lower valency. So probably you have that some of them are trivalent, some of them are divalent. Therefore, they will still probably partition within the condensates until they get degraded further and somehow contribute. So that's, of course, a simplistic picture that I showed in the next slide for this back of the envelope calculation. It's definitely the case that partially degraded constructs will still partake.
So any evidence then of vacuole formation in the living cells?
In the bacteria, you won't
Yeah.
be able to see they're too tiny. In the eukaryotic cells, there are strange things that you see, and you see sometimes literally big vacuoles, but I don't think it is easy to determine that that's due to degradation and what you know happens in vitro as well. I think there could be other processes.
Yeah, very interesting.
Yeah.
Thank you.
All right. Thank you so much. Oh, there's I think there's one more question in chat from Arpita.
Okay. No, I can't find the chat anymore. What is the chat? Oh, there. Arpita, thank you. A very basic question here. Do you think there are any differences between a droplet and a condensate? This is like a definition of sorts. I think people sometimes use the two terms interchangeably. I think a droplet probably implies a liquid state more than a condensate does. A condensate could be solid-like or gel-like, but I think condensate is perhaps more accurate, but sometimes people refer to these things as droplets as well.
Thank you. That reminds me of the discussion. What's the difference between a liposome and a vesicle?
Yeah. Yeah, I mean, I guess it's similar.
Yeah.
As long as we understand each other, I think both are fine.
Then we're good. Yeah.
Yeah.
All right. Thank you so much. Thanks for an amazing talk again. And thanks
you,
everyone
Kate.
for the discussion. And see you. Have a great rest of your day, rest of your evening, whatever it is for you. Bye.
Bye-bye. Thank you.